Namaskar, I'm Gunjan Bidani. As the whole world battles the coronavirus pandemic, every single day is bringing in hope. Hope of a breakthrough. Hope of a new vaccine. In this COVID-19 bulletin, we bring you facets of that hope, curated information on the new developments in science and technology each day that can help us in managing and overcoming the ongoing crisis. India and Israel have joined forces on the R&D front to battle the COVID-19 virus. According to the Office of Principal Scientific Advisor to the Government of India, both parties have discussed a joint R&D strategy for rapid diagnosis based on big data and artificial intelligence to enable a rapid return to routine life. In this partnership, the Defense Research and Development Organization or DRDO and Council of Scientific and Industrial Research or CSIR will work with Israel's Directorate of R&D in the Ministry of Defense. In this regard, Dr. Ron Malka, ambassador of Israel to India, tweeted, and I quote, "I am proud to connect brilliant minds from India and Israel so they can jointly develop life-changing solutions for the whole world and especially in fighting the COVID-19 pandemic."
I'm happy to share that the brightest minds, scientists from Israel and India, are jointly working now using advanced technologies like artificial intelligence and big data to find new tests, rapid tests for COVID-19 that will give results in few seconds. Hopefully we will have from them good news that will be life-changing. This cooperation is part of the successful cooperation that is there between Israel and India, and especially the decision of our two prime ministers to cooperate and work together.
CSIR's Hyderabad-based Center for Cellular and Molecular Biology or CCMB is executing a twin-module approach for tackling the COVID-19 pandemic. In this approach, the institute is trying to better understand the biology of the virus by its genome sequencing and simultaneously attempting to grow the virus in laboratory conditions. Genome sequencing of coronavirus strains from different locations of India will help us in understanding the commonalities and differences between various strains. This information will help in designing drugs against the coronavirus strains which are doing rounds in India. Apart from this, the information will help us to understand the genetic basis of why some strains cause more mortality than others, why some strains cause asymptomatic cases, and whether the sequence determines the transmission rate of a strain of the virus or not. The scientists are also attempting to culture large amounts of the virus with the goal of vaccine development. Let's hear more about this from Dr. Rakesh Mishra, Director CSIR CCMB.
Making large quantity of virus is very useful. You can use this virus inactivated and use it for vaccine. So we are interacting with the companies. We have finalized a project which is supported by CSIR, and we are going for inactivated virus vaccine. So if that helps, that will be wonderful. We are going to not only giving the virus for vaccination trial and scaling up, we are also going to test the properties of the vaccine from the patient serum to see if the vaccine is functional or not, so that it can go to for the trial purposes. The other use of large quantity of the virus is that it can be used for making the antidotes. For example, we have signed agreement with the company in Hyderabad that makes antidotes against snake venoms and other such things. So we are giving them the virus, so they will make antibodies against the coronavirus, COVID-19, and try that if that vaccine, that antibody kills the virus from the patient samples. If that happens, then they can look for permission to go and try straight away antibodies for the treatment for the patient. So this will kind of substitute or replace the plasma therapy that is being tried. In plasma therapy, we are using whole plasma of a patient who has recovered from the disease for a patient who is going through the difficult time, instead of whole plasma which comes with all other kinds of problems. This antibody treatment might be far superior to that.
In a collaborative research project, scientists from the Indian Institute of Technology or IIT Goa and Georgia State University will work together to find a potential drug against COVID-19. For such a study, the team at IIT Goa is planning to synthesize newly designed compounds and then check their bioactivity against the virus. Researchers will be using the available biological database curated by the international scientific community for carrying out this research. After identification, further stages of drug development will include preclinical studies, clinical studies, and other subsequent phases. Let's know more about this collaboration and work from Dr. Rishikesh Narayan, Assistant Professor at IIT Goa.
I have designed few organic molecules, so molecules, small molecules, which have potential to target coronavirus and sort of stop its spread in our body. So, or minimize its spread in the body so that body can take over and cure itself. So that's typically how these drugs work. So, first step of my research is the design. I have to design the molecules, which I have already done. And then the next step of our research is to validate these computationally, because labs were not open, labs are still not open. So I thought that in the meantime, I can do the computational validation of my molecules, that at least in computer, these are able to target coronavirus. So I have 3 collaborators from United States, different universities in United States. So I took their help. One of those collaborators is Professor Donald Hamilberg. He's at Georgia State University. So I took his help, and they did the computational validation. And so last week I got the results that these compounds, which I have designed, are good. And I mean, they are able to at least target coronavirus in the computer, computationally. Now, the challenge is we have to go to lab and synthesize these compounds in the lab. Then we have to do the biological evaluation of these in the virus itself. So there are 2 ways to do. You can also validate this in the test tube, and of course you can do that in the virus itself. So first, after the synthesis of the compound, we will take it to the in vitro validation, which is in the test tube. In any lab it can be done, or most of the lab it can be done. And then if the results look good, then we plan to take it further and do the in vivo validation, where we will do it in ready live virus and test them. So that is the plan.
That's all for today. We will be back with another edition of COVID-19 Bulletin soon. We hope that you all are safe and maintaining social distance. See you soon. Namaskar.